Now we will study about the cell cycle and its phases, biochemistry, cellular cell cycle and its phases, cell cycle phases and checkpoints. These checkpoints control transition between different phases of cell cycle, and there are regulators like are cyclins, cyclin-dependent kinases or CDKs, and tumor suppressors. M phase of the cell cycle, it is the shortest phase of the cell cycle. It includes mitosis like prophase, metaphase, anaphase and telophase, and cytokinesis. In cytokinesis, splitting of cytoplasm takes place, and G1 phase of the cycle is of variable duration. Regulation of cell cycle takes place by help of different steps. First step is cyclin-dependent kinases or CDKs, where they constitutively expressed by inactive when not bound to cyclin, and cyclin-dependent CDK complexes. Cyclins are phase-specific regulatory proteins. Their mechanism is activated by growth factors. Bind CDKs, phosphorylate other proteins. For example, RB to coordinate progression must be inactivated at specific times. And there are tumor suppressors. p53 pathway, p53 induction leads to p21 induction and CDK inhibition. Growth factor, there are different growth factors. For example, insulin, PDGF, EPO and EGF. They bind to tyrosine kinase receptor to transition cell from G1 to S phase. There are three cell types. One is permanent cell types. They remain in G0 phase or regeneration phase. They remain in G0. They regenerate from stem cells. For example, neurons, skeletal and cardiac muscle cells, and RBCs. Stable cell and quiescent cells, they enter G1 from G0 when stimulated. For example, hepatocytes, lymphocyte, PCT and periosteal cells. And stable cell enter G1 from G0. And labile cell, they never go to G0 phase. They divide rapidly with short G1. Labile cell, they divide rapidly and they have very short G1 phase. For example, the labile cells are mostly affected in the chemotherapy. The examples of labile cells are bone marrow cells, gut epithelium, skin, hair follicles and germ cells. Molecular pathways and interactions. DNA damage, it triggers p53. Apoptosis, that is intrinsic pathway, and back activation leads to caspase activation. HPV-E6 affects p53. Transcription, RB-E2F interactions regulate gene transcription for S phase.